Chapter 3, Section 11, chemolithotrophy and phototrophy. Chemolithotrophy, okay? Chemo means it's going to use chemical compounds for its energy source, but the lithotrophy here means it's not going to be organic compounds, nothing containing carbon. So it's going to be inorganic. Hydrogen sulfide, hydrogen gas, ferrous iron, ammonia containing compounds, or some variation of all of these. So glycolysis, not really happening here. Maybe citric acid cycle, maybe something else, or a variation of, because there's really no carbon-carbon strands to be utilized here. So they're going to use other mechanisms. Still going to somehow get those excited electrons, okay? Since they are chemolithotrophic, they're not using carbon-carbon, so they're not going for the sugars, the fatty acids, the amino acids. They're using other things, and they're using those to reduce NAD to NADH. Get those excited electrons, and then pass them along. Pump hydrogens. Set up their own proton motive force, which will then power ATP synthase. Proton motive force that you can see here in Ralstonia will then power all these flagella. Carbon dioxide with some energy, and you will be linked together to start forming the cell's constituents, its own amino acids, its own lipids and phospholipids and whatnot. Just because these organisms do not use organic compounds as their energy source does not mean they do not have carbon in them. They are still organic organisms, meaning they are based on carbon-containing molecules. Oxidative phosphorylation will occur, whether you're talking chemoorganotrophs, things that use organic compounds, kind of like us, or chemolithotrophs, inorganic. They're still going to have a variation of the excited electrons transported by NADs and FADHs, NAD and FAD, the bleeding of the energy by some cytochrome-like proteins, or cytochrome-like compounds, pumping the hydrogens, and then ATP synthase, making ATP. Just where their carbon is coming from. Carbon dioxide for chemolithotrophs, they'll link them together. Two carbon dioxides together gives you, a 2-carbon strand, acetate. Start linking more and more of those together. You can start building up sugars. You can start building up fatty acids. And from there, sky's the limit. For the chemoorganotrophs, their carbons that they're going to utilize are going to come from whatever organic compounds, proteins, nucleic acids, lipids, they take up into the cell. Phototrophy is completely different. Here, it's going to be light. Light exciting a pigment, just like you learned with photosynthesis, except here, if we're talking bacteria, some algae, it's not going to be quite the photosystem 2 and photosystem 1 that you learned about in Intro to Cell or plant biology. There are going to be pigments that get bombarded by photonic energy that will become, will excite an electron that will then be passed on to some form of an electron transport chain. In the process of passing on that electron, that excited electron, cyanobacteria, algae, plants split water, releasing the oxygen as a byproduct, taking that hydrogen, the hydrogens, and replacing the electron with those hydrogens, or the electron from those hydrogens. Other organisms, many bacteria, just the straight up bacteria, are anoxygenic. They don't do this. If they have some type of pigment in them, they are not being photosynthetic like you would think, where oxygen is the byproduct. Take an example right here. This purple bacterium, and I cannot remember the name of. No, I don't have it. But anyways, photons of light, photons of energy hit the pigments, excite the pigments. The pigments pass along that energy, give up an excited electron, Q'd QH2, which will then be cycled through this cytochrome complex, that high energy electron, boom, boom, boom. Since this is anoxidinic, that electron will then be passed back here. Notice, no splitting of water. That electron gets excited, gets recycled, and then passed back. In the process of taking that energy, bleeding that energy off that electron, hydrogens get pumped, setting up our proton motive force, and then those hydrogens flow back through ATP synthase. There's the ATP for that cell. So there is going to be a photosynthetic reaction center. Not the ones that you learned, like I said, intracellular plant biology, but there will be complexes, reaction centers that are very reminiscent of it. Just not with chlorophyll. It's going to be other things. Those photopigments, chlorophylls, bacteriophils, aren't always going to be green. Previous slide, purple. So that means purple is going to be the pigment of choice, or the pigment that reflects the purple light, I should say. Absorbs all others, reflects the purple. Just like plants absorb all except for the wavelengths that correspond to green, which is why we look at leaves and see green. They get, remember I just pointed out on that slide with the picture, what is referred to as cyclic photophosphorylation. That purple bacteria keeps recycling the same electrons over and over and over. Excites them, passes them on to the cytochromes, bleeds the energy, passes them back. Re-excites, passes them over the cytochromes, bleeds the energy, passes them back. You do not see this with normal respiratory organisms. Okay, there's going to be a final electron acceptor of oxygen, sulfur, nitrogen, or something like that. Yeah, I've already talked about many of this, so I'm not going to.